I wanted to begin by quickly thanking RECOMB for inviting us to give this talk and escape the Wisconsin winter for a few days. As we've already heard today, transcription is controlled at many different levels, and we're showing a few of them here. If we zoom all the way in, we can think about things such as DNA methylation, chromatin modifications in the DNA sequence itself. And then as we begin to zoom out, we consider transcription factor binding sites, as well as these larger regulatory regions, such as promoters and enhancers. And then as we zoom all the way out, we begin to think about these long-range chromatin interactions, such as the one shown on the left there, or your right. And we're going to focus on those long-range interactions during this talk. So as Joe mentioned earlier, chromatin structure is an important player in gene regulation. And we're showing a few different mechanisms by which this occurs. The second one there, for instance, looping, describes how the chromosome loops so to bring together distal enhancer regions and promoter regions nearby genes in order to regulate genes. So chromosome conformation capture technology allows us to directly investigate this, and I won't spend too much time describing the details, since Job already has. But the general way this works is there's a cross-linking step followed by digestion, ligation, and reverse cross-linking. And what this gives us is a map of the loci on the genome that are actually close together in three-dimensional space. So there have been a variety of technologies over the last 10 years or so which describe this. So 3C was the first. It allows us to investigate the relationship between one specific region and another specific region. Then we have 4C, which allows us to investigate the relationship between one region and every region genome-wide. 5C, which was mentioned earlier, which allows us to measure the relationship between many different regions and many other regions. And then finally, high C, which allows us to measure the interactions between every region on the genome and every other region. So given this data, we can ask a few different questions. First, we can ask, what are the functional implications of chromatin structure? What does this actually mean? How does this impact the function of the cell? Secondly, we can ask how this chromatin organization changes across different cell types or different tissues. And then finally, we can ask, to what extent is this chromatin organization conserved across different species? In order to answer all of these questions, we need systematic ways to analyze genome-wide high-C maps. The first step, and this is a pretty well-established process by this point, to analyzing this data is representing it in a form that we can do something with computationally. So we begin with a library of mapped reads to areas on the genome. And the first thing we do is we bin the genome at a set size, so at a set resolution, say 100,000 base pairs or 1 million base pairs. In our study, we used 350,000. And then we count the number of interactions, so the number of reads between each of those regions. And then we apply some statistical techniques. And based upon that, we can get this graph out at the end where the nodes represent all of the loci, or all of the regions on the genome, and the edges between them represent a significant interaction in three-dimensional space. There are a few challenges to doing this. It's not quite as simple as it sounds. The first one, and this was mentioned earlier, is that we just see more reads mapped between the nearby regions on a single chromosome. And we also see that there are more interactions, so more reads mapped between any regions on the same chromosome, as opposed to between different chromosomes. And then finally, even when we're looking at interactions between different chromosomes, there are some biases that make that challenging, such as sequence-based biases. In order to account for these, we can use different models for interactions between chromosomes and within the same chromosome. And then sort of a more subtle challenge, shown at the bottom here, is that these edges are not independent. So this has been referred to as the transitive property of chromatin interaction, kind of like that, whereby if we have an interaction between region A and B and region A and C, well, that means that they're sort of forming a clump in three-dimensional space. So it's more likely than chance that we'll see an interaction between regions B and C there. Okay, so what did we do? Well, we're given these high-C maps from many different data sets, and these come from a variety of different experiments. And we also have additional functional data. once again from a variety of experiments. And what we want to do is first cluster these high-C maps, and then based upon those clusterings, we want to analyze them for functional enrichment in different feature sets, and I'll talk about what exactly that means in a minute. And then we also want to analyze these clusters to see what sort of overlap there is from clusters in different data sets, whether those be from different tissues, different species, and so forth. The way that we do this clustering is using something called the Markov cluster algorithm. And this is an algorithm that's designed for clustering large, sparse graphs. It uses a stochastic flow-based approach to doing this. There's one main parameter that we have to select as part of our clustering, and that's the inflation parameter here. And in order to select this inflation, what we do is we try a bunch of different values, and then we select the one that minimizes this index shown here, which is a variation of the Davies-Bouldin index. The main idea here is we're looking for a clustering that produces a good number of clusters, and the quality within those clusters is pretty good, but we don't want too many clusters, and we don't want too few clusters either. We applied this approach to data from a recent paper in Saccharomyces cerevisiae, and we got this clustering where we have 3 really large clusters shown in this bar graph, and the different colors here represent regions that are coming from different chromosomes within yeast. So you can see each of these 3 clusters contain regions from a variety of chromosomes, and when you combine those 3, they account for about 80% of the yeast genome. We can zoom in on those smaller clusters down at the bottom as well, and even though they have many fewer regions, we can see that they still have regions from a variety of different chromosomes. Here in this Sarcos diagram, what we're showing is the actual interactions between different regions which are contributing to these clusters. So this is genome-wide, all of the interactions between clusters, the 3 largest clusters, as well as the other ones in different colors. The different bars on the side represent different chromosomes, and it's tough to sort of tell too much just from looking at this. So we can zoom in on the individual clusters. So this is the largest cluster at the top right here, the second largest, and the third largest. So as we discussed before, you can see there are regions from a bunch of different chromosomes in each of these. But at the same time, you can see these clusters actually refer to different areas of the genome. You can see in the bottom right, for instance, that it's pulling a lot of areas from that looks like chromosome 4. And the blue one has some of those as well. So this is actually separating out the genome pretty well. Given these clusters from these different high-C data sets, the next question that we wanted to ask was what were the functional implications of these? So the way that we do this is as follows. We take a set of regions corresponding to some feature. So I'm showing a few different types of features that we take data from here, such as TF binding sites. So for instance, in the TF knockout experiment, we can take the set of genes that were regulated by each of the transcription factors measured in that experiment. We can map those genes to the regions nearby them on the genome. And based upon that, we just get out a set of regions. And we can measure the overlap between each set of regions and the regions in each of the clusters from our experiment. That gives us a p-value, just a standard hypergeometric p-value. We create a background distribution of p-values for random clusters overlapping with random sets. And then based upon that, we determine the likelihood of seeing that size of overlap between the cluster and the set of regions. And then we correct for multiple testing. We set a threshold, and based upon that, that tells us what sorts of feature sets each of these clusters is interacting with, or each of these clusters is enriched for. So we applied this to our yeast data for each cluster. And what you can see here is a variety of different feature sets which are enriched for one or more of our clusters. These include things like origins of replication, evolutionary breakpoints, centromeres, tRNA sites, all of which have been established before as being enriched for interactions in 3D. And in addition to that, we established that the targets of a number of transcription factors are also enriched in one or more of these clusters. And the transcription factors I'm showing here are involved in things like chromatin remodeling and replication, so these are pretty important. Having established that our method can recover known features in yeast, we wanted to ask, how is this structure conserved between different tissues. So to do this, and the example I'm using here has stem cells and cortex cells from mouse. What we do is we cluster each of those data sets. And then just like we looked at the overlap between the clusters of regions and the sets of regions for each feature set, we can look at the overlap between each of the clusters here, since the library of possible regions for each set is the same. So same approach here. We see the size of the overlap, get a p-value from that. We have a background distribution based upon random clusters. We correct for multiple testing, and that will give us a set of clusters that are significantly overlapping. We applied this to mouse data, so stem cells and cortex cells from a recent paper. And what we see is the distribution of clusters here is actually pretty different than what we saw for yeast. So instead of having a few large clusters drawing from a bunch of different chromosomes, we see in the case of stem cells, we have one really big cluster with regions from a bunch of different chromosomes. And then in addition, we have a bunch of smaller clusters, primarily drawing from one chromosome. In cortex, we see a pretty similar distribution. And we applied this to human tissues as well, HIMR 90 and stem cells, and see fairly similar results to the cortex cells. We compared the overlap between these clusters in each of the tissues, and the blue boxes here represent significant overlaps. So what you can see is that first, a large amount of this structure is conserved. We found that about 78% of the regions co-cluster between cortex and stem cells. So a lot of this sort of very high-order structure is conserved between the two tissues. And then you can also see that for the most part, these are one-to-one relationships here. So while there are some clusters that overlap more than one different cluster, this sort of indicates that these clusters are pretty similar. And what I'm showing on the right is just some gene ontology processes, which were enriched for some of these clusters that were overlapping. Okay, so the final question we wanted to ask, once we'd established that there is this overlap between tissues, is can we determine some sort of overlap between clusters from different species in the same tissue? And the way that we do this is pretty much the same as between different tissues. The only complication is that the regions are different in different species. So we map the regions to genes, and then we use known gene orthologies to map between the two. So we apply this to stem cells from mouse and human. And what you see here is, once again, there's extensive conservation, not quite as extensive as in the case of different tissues, but still, it accounts for a large amount of the large clusters in particular. So in conclusion, we produced a new graph-based clustering approach for analyzing high-C data. This generalizes across different species, so it can be applied to yeast, human, mouse, or other species as well. It enables comparison across different maps using this clustering approach, which makes that a little simpler than some other possible methods for doing it. We recover known and novel features of yeast chromatin organization. We show that high-C clusters have significant overlap between different tissues in the same species. And finally, we show that in comparable tissues, clusters are conserved between different organisms. Thank you, and I wanted to acknowledge the following people for helping me prepare this talk.
Thanks, Robert. Time for one question only.
I can't take the only one, so you have to have two or...
Okay, two questions.
I'm sorry. All right, quick question. So your clusters show functional enrichments, and this is an unsupervised clustering approach. My question is, if you took a supervised learning approach and you said, can we now look for things that sort of can recover these features? Because I noticed some overlap in your functional enrichments between clusters 1, 2, and 3. My question is, if you now took just each of these functional categories and you try to build a supervised approach to recover them, do you think you might have more power to distinguish them?
That seems like a good idea. It's something that we should definitely look at. I'd have to think a little more about it to
Thank you.
be able to give a better answer.
Hi. So before you obtain your clusters, you have to bin these chromosomes. And how robust are your results to the scale of the binning? And are they also robust to how do you decide on that scale? And are they also robust to translating these bins?
It's a good question. We tried a few different resolutions for the high-C data in the higher eukaryotes, so mouse and human. And we settled on 350,000 because first off, we were analyzing data from different high-C experiments, so we had to sort of pick a happy medium where there was good enough coverage in the data in all of those cases, but still detailed enough for our purposes. We looked at as tight as 100,000, as wide as 1 million, settle on it for that reason. However, our method for comparing between different species can actually be used for comparing between different resolutions as well, since we map to genes, and then we can look at the overlap between genes. So that is one way to use that for comparing different resolutions and sort of determining how that impacts the results you get.
Okay, thanks a lot.